This video will cover the process of cell division. The cell cycle has two distinct phases, interphase and the mitotic phase. All chromosomes are duplicated during the S and G2 phases of interphase. The two identical copies of each chromosome are called sister chromatids. There are some key terms to understand in cell division: double chromatid and single chromatid chromosomes, chromatin, homologous chromosomes, and chromatid. The steps of mitosis include interphase, prophase, metaphase, anaphase, and telophase. Note that interphase is not technically part of mitosis itself. During interphase, the nuclear envelope is intact, and chromosomes are duplicated but not yet apparent. DNA is loose, uncondensed, and called chromatin. In prophase of mitosis, double chromatid chromosomes become evident as chromatin supercoils and compacts. The nuclear envelope and nucleoli disappear, and centrioles migrate toward the poles, forming the spindle. This image illustrates double chromatide chromosomes, which are formed after DNA duplication. During metaphase, double chromatid chromosomes line up on the equatorial plate. Spindle fibers attach to the chromosomes at the centromeres. Anaphase begins as centromeres are pulled apart. Groups of single chromatid chromosomes move to opposite poles of the cell, pulled by spindle fibers. In telophase, single chromatide chromosomes reach the poles. The nuclear envelope and nucleoli begin to reform, and cytokinesis starts, forming two new daughter cells. As the cells return to interphase, cytokinesis completes and chromosomes disappear as interphase chromatin reforms. The two new daughter cells enter the G1 phase of the cell cycle. This summarizes the process of mitosis. Typical body cells, called somatic cells, divide continuously through a process called mitosis. The new cells receive an exact copy of the parent cell's DNA. Somatic cells are diploid, or 2N, meaning they contain the full number of chromosomes in pairs. Humans have 46 chromosomes, or 23 pairs. Mitosis occurs only in somatic cells. Sex cells, called gametes, are not diploid, they are haploid, or N. Gametes are eggs and sperm, produced in ovaries or testes. Our haploid number is 23, so our eggs and sperm each have half the number of chromosomes, 23. Fertilization is the union of an egg and a sperm. If both were diploid, the resulting fertilized egg or zygote would have double the necessary chromosome number. Instead, gametes are haploid, so at fertilization, N plus N equals 46. Both egg and sperm have exactly half the number of chromosomes as somatic cells. Mitosis and meiosis differ in the number of divisions, the resulting number of chromosomes, and the number of products. Meiosis is the cell division process that transforms cells from diploid, 2N, to haploid N. Somatic cells in the ovaries or testes must produce haploid gametes. Meiosis is more complicated than mitosis because gametes must contain precisely half the diploid number, specifically one of each homologous pair of chromosomes. This is a representation of a human karyotype, showing the pairs of homologous chromosomes. In males, the 23rd pair of chromosomes, the XY pair, is not homologous. All other pairs of chromosomes are homologous. Meiosis has two divisions, called meiosis I and meiosis II. Meiosis starts with one pair of homologous chromosomes. DNA is replicated. Meiosis I reduces the cell from 2N to N. Meiosis II separates chromatids into 4 products. This image provides an illustration of the process of meiosis. In males, typically 4 viable sperm are produced following each meiosis II. In females, only 1 viable ovum or egg is produced, along with 3 small polar bodies. Crossing over occurs only during prophase of meiosis I. Homologous chromosomes temporarily join in tetrads, overlap, and trade DNA, promoting genetic diversity. Meiosis I involves DNA replication, and the parent cell produces 2 haploid daughter cells, each with 1 member of each homologous pair. Crossing over may also occur. In meiosis II, there is no further DNA replication. Chromatids separate, and each daughter cell divides, resulting in 4 daughter cells, each with half the number of chromosomes.